So I think we'll get started now.
Mm-hmm.
So welcome everyone. I'm Paul Danis with the consortium. Glad to have you join us today for an exciting day. Let me just give a quick couple intro slides, and then Brian Drown will be moderating the rest of the day. So first, you know the title, the topic is exciting. Can't wait to hear the story about that. I just first want to thank our annual sponsors, enable us to do this kind of work: Thermo Fisher, Bruker, SCIEX, and Lilly. Our major sponsors, as well as our supporters, Agilent and Pfizer, and a new supporter just came online as Mass Spec Consulting. So we're very grateful for the support that they have provided to us, allow us to do these kinds of events and get together and talk about important things in our arena. The big news, of course, you may have seen the emails that we have our third International Top-Down Proteomics Symposium going to be in Hamburg, Germany next summer. Details are still being worked out, and we'll have that information for you shortly, but you can pencil that in on your calendar and start making plans for a great festival of proteoforms and top-down proteomics. It's going to have a range of topics, as you can imagine, things that'll probably engage and excite most of you. And we'll have information on logistics, abstracts, registrations, and all those types of things pretty shortly, and we'll get that to you as soon as that's available. So I'm looking forward to that, and looking forward to seeing a lot of you folks there. Just as far as speakers for this event here, we typically just invite people that look like they have interesting work. They're doing a lot of cutting edge research, a lot of early career people. So if you have some interest in presenting, or if you know someone who would be a good presenter for this, or someone you'd like to hear their story, just send us a note, drop us a note in the chat. You can drop a note or send an email to office@tdprot.org, and we'll be happy to see about having some more of these folks that you recommend to come present. So with that, I'll hand it over to Brian, who's going to moderate today's seminar, and looking forward to hearing the story, Oliver. Thank you.
Thank you, Paul. And yeah, really excited to see that we're going to have our third meeting of the community upcoming next summer. But today is all about Oliver Hale. And so Oliver, he's a senior postdoctoral researcher in the research group of Professor Helen Cooper at University of Birmingham in the United Kingdom. Oliver, he completed his bachelor's degree and PhD in chemistry at University of Reading. In the Cooper lab, he specialized in native ambient mass spectrometry, which is a combination of native mass spec, top-down mass spectrometry, and ambient mass spectrometry imaging. And this has enabled proteoform and complex characterization being analyzed directly from tissue. And so his work is applying this approach as a molecular pathology technique for studying neurodegenerative diseases involving proteinopathies. And so I think we're going to hear a little bit about all of that today. And so help me and welcome Oliver to tell us about his work. Thank you, Oliver.
Thanks, Brian. Right. Let's get my slides going. That look good?
It sure does.
Okay. Okay, yeah, thank you for that nice introduction. Thank you for the invite to speak here as well. So I'm going to talk about the kind of technology as we've been developing it in the last couple of years to measure, to analyze intact proteoforms directly from tissue with mass spectrometry. And then I'm going to talk about an application which we are very excited about, and it's kind of the future of this technology, I think, is in applying it to these kind of proteinopathies. So I want to talk about molecular pathology. So this is essentially a set of techniques that allow us to understand disease mechanisms at a molecular level. And this is techniques like spatial biology technologies that allow us to map molecules in tissues. And this is extremely important because what the molecules do in a tissue is it's governed by everything else that's in the tissue, so the cellular environment and that kind of thing. So when we're looking at a protein or proteoforms or their complexes, we need to understand how they interact with everything else in that cellular environment. And there are techniques to do this, spatial biology techniques like immunohistochemistry, but this is a targeted technique, so it's antibody based and requires you to know something about your sample prior to analysis. And then there's mass spectrometry imaging techniques like MALDI, but this is typically limited to smaller molecules like lipids, but it's untargeted, so you can use this as a true discovery technique. So existing spatial biology techniques lack proteoform and complex level information, and so we need techniques which deliver this information, because otherwise we will miss these interactions between metal ions and small molecule cofactors and proteoform interactions. And it's critical to know how these processes work, because the function of a protein or their malfunction in a disease is related to that, to those interactions and their structure. And mass spec imaging is one of those techniques that's really kind of built for this kind of work. It's multiplexed, it's untargeted, and it's extremely sensitive, and we can get very specific information out of it. And the concept is very simple. The intensity of analytes, say 3 different proteoforms, differs across a tissue section. And by mapping those intensities to the tissue section, you can build up a map of those proteoforms. And of course, their location is then important to the disease pathology if you were studying a disease. And if we could combine that with native mass spectrometry, which allows us to look at folded proteins and their complexes in the gas phase, we could actually study complexes within the cellular environment of tissues. And so native MS combined with imaging would be really fantastic. And that's what we've been striving towards in the last few years, because of the number of diseases that are associated with misfolded proteins and irregular protein ligand binding. So under the umbrella of NAMS, native ambient mass spectrometry, we've been developing the combination of native mass spectrometry, so taking intact protein complexes and analyzing them in a gas phase, top-down mass spectrometry, where we take the intact protein ions, we fragment them to obtain sequence information about their post-translational modifications and their identities if we do not know what they are in the first place, and ambient mass spectrometry imaging. So this is untargeted spatial analysis, and it's important that we use one of these ambient MS techniques because they are electrospray based, and that gives us compatibility with the highest performance mass spectrometers like FTICR, Orbitrap, QTOF, and allows us to analyze very large analytes and also get the best top-down performance. And this comes together in a technique which we call NAMS to look at things like ligand binding in situ, obtain sequence information, stoichiometry, PTM information, and metal ion binding information, all with a single technique. And originally we used technology called LISA, and so this is liquid extraction surface analysis using the Advia Nanomate robot, and that's a commercial system that you can still buy today, and it's a pipette-based system where the non-denaturing solvent system is applied to a tissue surface with a pipette tip. It's then extracted, taken to a chip-based nano-electrospray emitter, and sprayed into the mass spectrometer. And the first demonstration of this was analyzing dried blood spots. And for example, here you can see hemoglobin tetramers bound to 4 heme groups intact in the mass spectrometer. And that moved on eventually to native mass spectrometry imaging with LISA. So you can see proteins from 14 to 43 kilodaltons here. And in fact, this image here is the RID-A homotrimer. It's 42.6 kilodaltons. It has a spatial distribution which you can see at low resolution with LISA, and it was kind of the first demonstration of these complexes beyond hemoglobin. But obviously, the spatial resolution of these images is not that high, and really to be taken seriously as an imaging technique, you need to be approaching single cell type level, and certainly with higher resolution than LISA allowed. So we moved to using nanospray desorption electrospray ionization, or nanodesi. And it's not commercially available. It's a home-built ion source, but it offers much higher spatial resolution than LISA, but it offers the same concept. So it's a physical liquid junction that makes contact with the tissue. It's the same concept as LISA. So it was a straightforward kind of next step when we made this jump. A non-denaturing solvent system is fed through this first capillary to the tissue surface where a liquid junction forms, and your analytes, including any proteins or lipids or small molecules in the tissue, are desorbed into this droplet, and then they are aspirated to the mass spectrometer inlet where they're ionized by electrospray ionization for analysis in the mass spec. And this is a photograph of the ion source we use. This is on an Orbitrap Ascend mass spectrometer. You can see it's quite a small ion source. It's readily removable, portable. This one is actually the one we built in Birmingham, and this is actually in San Jose in California. So it's very transportable. You can see here these are the 2 capillaries. So this first capillary is delivering the solvent system to the surface, and then the second one is aspirating to the mass spectrometer. And you can see just in this photograph, you can see the liquid junction just about here. It's approximately 90 microns across here, so good for imaging at about 100 micron spatial resolution. And what this has enabled us to do is to push to higher resolution imaging. And so these are just two protein complex examples. So this is GTPase RAN plus its endogenous ligand GDP plus its magnesium cofactor. You can see it's most abundant in the cerebellum of the brain. And the carbolic anhydrase plus zinc complex, you can see that's more abundant in the corpus callosum of the brain. And so this slide just kind of shows when we did nanodesi of the kidney again, and showing, so for example, the RID-A, we have the distribution in the cortex here, and the HFABP is in the medulla here, and another complex right in the renal pelvis region. And what it's possible to do is not just look at cations, but also anions here, and that turns out to be really useful, which I'll cover in the next couple of slides. So here's a brain section. Here's a protein with a distribution within the brain. It's it's most abundant in the front of the brain and less abundant at the back. And when we went to identify it, we obtained nice B and Y fragment ions. The B ions revealed that there's a covalent modification to the N terminus of this protein, myristoylation. And we also end up with this strange neutral loss pattern here. And when we look at the ARF3 intact molecular weight, we actually notice there's a molecular weight difference of 443 Daltons here. And that kind of corresponds to this molecular weight shift here. And that corresponds to the molecular weight of GDP, so this ligand. But in the positive mode, it's not possible to directly detect it because it doesn't leave with any charge, it leaves neutral. So if we flip to negative mode and do the MSMS in negative mode, we actually get a charged loss of the GDP, resulting in a loss of a single charge from the precursor. But that means we can actually detect the ligand intact after MSMS. And because we're using a tribrid platform mass spectrometer, we can do MS to the N. So we can do MS to the 3 here and fragment this ligand. So we obtain not just the intact molecular weight of the ligand to identify it by, but also we have a fragment ion spectrum, which can be then assigned to the fragments of the GDP. So we get not just intact molecular weight confidence, but we get fragment-based confidence of ligands, and we also have sequence information from the positive mode, so we can be extremely sure of our protein identifications when they're characterized in this way. So we also worked with membrane proteins, and this is an ongoing project actually, but was first demonstrated with eye lens tissue. And the cool thing about the eye lens is it has a really abundant membrane protein complex in it. It's called aquaporin 0, and it's observed as a perfect test bed for this work. So this is inspired by the work of Carol Robinson's group and their membrane protein analysis with nanoelectrospray ionization. We took concepts from that and applied it to nanodesi. So the solvent system has a detergent with detergent micelles in it. And the detergent solubilizes membrane proteins and encapsulates them within the micelles. And during electrospray ionization, we can activate those micelles to remove them from the membrane protein and end up with bare membrane protein ions for mass analysis. And you can see in this example here, so with the detergent, we see the aquaporin 0 signals, and without the detergent, we don't have them at all. And it's relatively straightforward to deconvolute these signals to obtain the intact molecular weight of aquaporin 0 here, 113 kilodaltons. But it's not always possible to figure out which charge states are present. You might only have one abundant charge state for a proteoform in the tissue, at least if it's low intensity. So we use another technique called proton transfer charge reduction. And again, it's a gas phase technique. We introduce the protein ions get trapped in the ion trap of this mass spectrometer, and we transfer negative reagent ions also into the ion trap, and the reaction between the cations and the anions reduces the charge of the proteins. And so these are artificially charge reduced. They're not the charge states that were generated directly from the tissue. They are the 17 plus precursor charge reduced in the gas phase. And what this means, that we can then deconvolute these signals to give the intact molecular weight experimentally, not just through deconvolution here. And if we want to know more about the complex, we can fragment it. So by isolating the 16 plus here and then collisionally activating it, we end up with this sequence of ejected monomers from the intact complex. And you'll notice that these are exactly 1/4 of, well, the subunits are exactly 1/4 the molecular weight of the intact complex. And so we know that the stoichiometry of that complex is 4. So there are 4 subunits in that complex, which is exactly what we'd expect for aquaporin 0. And we can go even further again with MS-3, and we're obtaining sequence information for this membrane protein directly, sampling it from the tissue, and obtaining the sequence coverage of approximately 11% here, which is actually pretty high for a membrane protein. And the most abundant fragments are always these fragments that occur at the aspartic acid residues. And actually, this sequence only has 2 aspartic acid residues in, and they are always the most abundant fragments. And of course, really, the main aim of this is to do the imaging, and the imaging aquaporin 0 turns out to be really interesting. So it actually has concentric rings, and this lines up with immunohistochemistry of rat eye lenses as well. So this is actually a sheep eye lens, but rat eye lenses also show this with the IHC. And we also found that there was a phosphorylated proteoform of aquaporin 0 that ended up with a different spatial distribution. So it actually fills in the gap when it's phosphorylated. I think that's really interesting. We don't have an explanation for it, but I just think that's amazing that we can look at the different proteoforms at 113 kilodaltons and see that they are mapped differently. And optimizing for membrane proteins doesn't actually exclude soluble proteins. So we can still look at these too. And the lens is full of crystallins, which form big complexes. And so these are 2 other complexes within the eye lens, beta-crystalline heterotetramer featuring 3 different subunits, and this beta-crystalline homotetramer featuring 4 subunits and then 2 ligands. And one of the fascinating things about the eye lens is the proteins at the nucleus are as old as the organism. So the proteins at the center of the nucleus are as old as you are. Whereas at the very edge of the eye lens, they are new proteins, newly generated. And so we have this separation of the protein, not just within the space of the eye lens, but also within time. So we know that this beta-crystalline heterotetramer is a newer protein, it's been newly produced compared to the beta-crystalline homotetramer. And I think that's really fascinating from a disease perspective, because we could start looking at things like cataract formation and the way PTMs are involved with that. And for example, UV light damage and causing different PTMs. So that's a work in progress, but I think that's really quite exciting. We've pushed recently to increase the molecular weight range we can address even higher, and we've managed to more than double it recently. So we've swapped from Orbitrap Eclipse to Orbitrap Ascend platform for this work. And we still have the issue of, okay, we got a complex mass spectrum. We don't know what the molecular weight of any single peak is, but by using PTCR again, we can assign this protein, and we know that these other signals are signals of that protein, but also we can deconvolute the PTCR mass spectrum to give an intact molecular weight of 231 kilodaltons. Now, this is pyruvate kinase, and it's currently the largest protein we've managed to analyze with nanodesi, and it's also been possible to image it. So this is just the back of a mouse brain. We have the we have the arbor vitae in the cerebellum and the cranial nuclei in the brainstem. And so GPI 125 kilodalton homodimer, that's a relatively homogeneous distribution. But if we look at the GAP-DH, this 143 kilodalton tetramer, it actually has a very specific distribution to the arbor vitae of the mouse brain. And what's interesting is we have these 2 images and they look the same. And the difference between these peaks is actually the same as the substrate for this, for the GAP-DH. And we think what we're seeing is binding of this substrate. We still have to confirm that, but that's entirely possible. LDHB, again, it has a different distribution. It's also in the brainstem as well as the cerebellum. And then the pyruvate kinase, the largest protein that we've been analyzing in imaging, also has this distribution in parts of the cerebellum. And I think it's worth pointing out, so mass spectrometry imaging, especially say with MALDI, is typically used for molecules less than 200 daltons. This is a 100-fold bigger complex or molecule than is typically imaged. So MALDI is typically used for lipids, really. I think that's really quite a selling point of this technique is just how big we can go, but also we can characterize these complexes. So I want to kind of like, so I want to move on from the technology development. I think I've shown how far we've come with it, but I want to now talk about its application to molecular pathology. And in particular, this technology, I think, is suited to proteinopathies. So this is diseases where changes to a protein or irregular metal binding or something like this affects the protein function, and that disrupts the cell function and causes the disease. And I think it really is suited to that application. And so I want to focus on amyotrophic lateral sclerosis. And this is a neurodegenerative disease. It affects the central nervous system, and it typically affects the motor neurons of the lumbar spinal cord and progresses up the spine, resulting in gradual paralysis until it reaches the brain, where you get the most severe symptoms, and ultimately it results in death. There is no better prognosis than that. The diagnosis is not one you want. And in the worst cases, it occurs in the bulbar region of the brain, and the onset is very quick, and you have immediate problems with swallowing and breathing and that kind of function. So it's a really terrible disease, and there is no therapy for this right now that has any improvement to the prognosis. So we've been focusing on a small proportion of the ALS cases. So it's only 2% of ALS cases, the familial, the inherited cases. So these are these are because of a protein called SOD1, and mutants of SOD1 have what's described as misfolding, and that's a very loose term. The canonical view has been that you have the SOD1 dimer, and it's misfolded in some way because of a PTM or because of a mutation, and it falls apart to monomers, and then those aggregate and you get inclusion bodies, and the disease progresses. But one thing that was also suspected about SOD1, or the kind of known but not its impact not known, was the number of metals that it binds. So it should bind 4 metals in each dimer, and the zinc ions are important for the structure of the protein, and the copper ions are important for the function. So they're their catalytic center. But it is possible for the SOD1 to not have the correct number of metal ions, and so we began looking at this, and in particular because the limitations of existing techniques mean that it's not possible to investigate this with these techniques. So there was an antibody developed for copper deficient SOD1 complexes, and when it's applied to tissue, there's nothing that distinguishes it from a control. It very, very heavy background staining, so it's not actually useful for in tissue work. It works as an ELISA, but it doesn't work in tissue. It's not specific enough. We could think about other mass spectrometry imaging techniques, and a lot of people have, but the problem with things like ICP-MS is you destroy the protein metal complex information. And the alternative, say with moldy MSI proteins, which is possible, you don't retain any of the metal binding information. So neither of those are actually suitable for this kind of work. And so NAMS comes in as the best option for this, because we can retain protein metal complex information into the gas phase, and also map the distribution. So we've been working with wild type and mutant mouse models. So our controls have been the SOD1 human wild type, whereas the disease phenotype is the human G93A model. And these are really easy to distinguish from a wild type mouse. I know, because we accidentally got sent some wild type mice, and I I confirmed that it was in fact wild type mouse with this B ion here, but it's really, really straightforward. And the mutation of G to A gives us a mass shift, which makes it really obvious when we're looking at the human G93A or the human wild type. Key here is that we were looking at these mice at 120 days, and there's a really severe phenotype at 120 days, and really real evidence of motor neuron loss, which I'll show in a second. But initial experiments by using nanodesi in the mouse brainstem revealed that we really did have this huge population of metal deficient species of SOD1 in the brainstem, especially compared to the control, where there is some, but the abundance is so much higher in the in the disease phenotype. And when these are deconvoluted into the mass space, you can really see that the difference here is really quite huge. And it's also worth noting that we have these glutathione-related dimers of this G93A complex as well, and we'll see images of those later on, and I'll explain the significance of them. So this is the workflow we had after the initial work. So a complicated slide, but there's two things I want you to take away. So the first one is that we're doing Nissl staining of serial sections with these tissues as well. So this is so we can count the motor neurons in these tissues, so that we know that there is a disease pathology that corresponds with whatever we find here. And then the mass spectrometry imaging here is selected ion monitoring type experiment where we're using a very wide window, but it helps with sensitivity and stability of the signal, essentially, to do a sim type experiment rather than a full scan type experiment. It's still a very wide window, but it's selected to some extent. So I just want to show the spinal cord especially is a real nightmare to work with. So this is a mouse spinal cord, and we're interested in essentially these two regions here, the lumbar enlargement, because this is where the disease begins. And these are fresh frozen, so they're very delicate tissues. They're not fixed. We have to work with fresh frozen tissue because otherwise proteins are disrupted or say cross-linked, and we can't analyze them in that way. So this is a mouse spinal cord section on a normal glass slide. So this is approximately 2 millimeters across, and it's extremely difficult to work with. But we've been successfully imaging them for a few years now, actually. And you can see, so just this top line here, this is RF1. This isn't relevant to the ALS, but it's a marker protein we're using to confirm that our experiments are consistent across days and weeks. But from the ALS perspective, what you'll notice is that where there is metal deficiency, so that's 2 or 3 metal ions, in the controls, there is no obvious spatial distribution, whether you're looking at the metal replete complex or the deficient complex. When you look at that in the G93A tissues, there's a real obvious specific localization of metal deficient complexes to the ventral horn of the spinal cord. And the ventral horn is where the disease occurs. That's where the pathology is. That's where the motor neurons degrade. And so when we first saw this, we were really quite excited. And what you'll see is with the metal replete complex, there isn't that localization. It's much more homogeneous. It has a lower abundance in the gray matter generally, but it doesn't have that spike in intensity in the ventral horn. And this is true for the monomers as well. The metal deficient monomers have the same distribution as the metal deficient dimers, and whereas the metal replete monomers have the same distribution as the metal replete dimers. And so this is really obvious when we look at the intensity of these signals. So in the ventral horn and the dorsal horn, so that's the ventral horn is where the disease pathology is, and the dorsal horn is where it doesn't occur. Comparing these two, you can see in the wild type, they are the same distributions, whereas in the ventral horn and the dorsal horn of the G93A model, there is this huge difference in the abundance of metal deficient monomer and metal deficient dimer compared to the dorsal horn. And so the dorsal horn we're not expecting to be affected by the disease, whereas the ventral horn is where the pathology is. So this is where we're seeing the change. And importantly, it corresponds to a significant change in the number of motor neurons in the ventral horn as well, when we study that with the Nissl staining. And again, I just want to highlight the difference here. So we're seeing this metal deficiency here, but where the immunohistochemistry wasn't able to pick it up. So we also study the brains, and the brains are very interesting as well, because we have a kind of different profile of them. We're sectioning them sagittally, so on the side. So what we get is this profile through the brain. So we can see the brain stem here and the front of the brain here. And so that gives us a profile through all these regions here that you see lighting up. And these are in the metal deficient, for the metal deficient complexes, really bright and abundant with specificity, whereas the full metal bound holodimer, the correct form of the protein, does not have those same distributions. It's most abundant in these ventricles of the brain where the cerebrospinal fluid is. One odd thing we noticed was in the controls, the metal deficient dimers are actually quite abundant in the hippocampus, and that isn't true in the disease model. And we actually don't have an answer for that right now. And pathologists we talked to don't have an answer for that. So it might be a quirk of that control, but so far we don't know why that is. But nevertheless, what we're seeing is this, the G93A SOD1 in a metal deficient state is abundant in these motor associated regions of the brain. And in triplicate, we see at different depths in 3 different brains, we can see how these regions light up. So we've got things like the hypoglossal nucleus. That's a region associated with things like swallowing. Facial motor cortex is things like smiling or coughing. And then there's other motor nuclei as well, which are responsible for many of the other facial muscles. And it's true that the metal deficient species are all localized, whereas the metal replete forms are not. And you'll notice here I've included the glutathione-related forms of the SOD1 complexes. And one of the hypotheses was that a big PTM like glutathionylation would disrupt the dimer interface and cause the complex to dissociate to monomers and then aggregate. But that doesn't actually seem to be the key factor here. It seems to be the metal binding is the critical thing, because whether or not it's glutathionylated, if it's deficient in metals, it has the same localization as if it's not modified. Whereas the 4-metal form of the glutathionylated dimer has a similar distribution to the unglutathionylated dimers. And this is even more obvious in the high resolution imaging of the brainstem down here. So we can see whether SOD1 is just N-terminally acetylated, it has this localization into the motor nuclei, and it's the same for the S-glutathionylated form. It's again in these motor nuclei in the same distribution as if in the unmodified form. Yet when we look at it with the holodimers, they are essentially homogeneously distributed with an abundance in the ventricle here. We also looked at the cerebellum, and the cerebellum isn't typically studied in ALS because it's not so associated with motor function, but there is some motor function associated with the nuclei in the cerebellum. And so here we have 2 marker proteins here, just so we can mark out the features of the cerebellum. So this is GTPase RAN and carbonic anhydrase again. But you can see with the metal deficient dimers, how they're localized to this gray matter, the nuclei here, the deep cerebellar nuclei, and whether or not they're glutathionylated or not. But this is the only time we see a difference between the glutathionylated form of the complex. So actually we see that with the holodimers, the 4-metal form without glutathionylation is pretty homogeneous, and with glutathionylation, it's actually really abundant in the molecular layer. And again, this is something we don't have an explanation for, but it's an interesting difference and on the proteoform level. So I think that's really exciting and a potential route for more investigations in the cerebellum of ALS mouse models. So where are we now? We've been working for a bit on ALS human cases. So I just want to give a slide on this. And so obviously, if we're looking at the familial cases, we're ignoring 98% of cases. So we've started looking at the human cases where they're sporadic, so the non-genetic cases. And we have been doing some imaging, some top-down and that kind of thing. And for example, just as an example here, we find localizations of certain proteins. So this is the S100A6 dimer, and it's a protein that's associated with neuroinflammation and oxidative stress. And so perhaps we'd expect to find it there, but it's a localization that we can start working from. And then the other side of things is looking at SOD cases in the human disease. And I think one of the fascinating things so far is we haven't found any metal deficiency in human cases. Now, I think that's because of the mutations we have. We've had mutations like E100G, which are even further along the amino acid sequence than the metal binding is than G93A. But what we have been finding is we get these heterodimers forming. So this is the signal for the E100G homodimer. This is the homodimer for the wild type in the same tissue. And then this is a heterodimer that forms between them. And it's within this pathology region. And work by others previously has linked the heterodimers to disease severity. So we're very excited to be able to investigate that in more human cases because we think there's a possibility it will have a spatial distribution which we can map within the tissue. So I just want to conclude by showing saying that we've got a uniquely capable molecular pathology tool with NAMs. So we can look at proteins up to 231 kilodaltons so far. We can identify the proteoforms and ligands that we're looking at and map them within a variety of tissue types. And specifically with respect to ALS and its molecular pathology, we've been able to provide new information on the metal deficiency of SOD1, but with spatial context. And also kind of suggest that some of the PTMs and monomerization hypotheses are not actually what's important here, but that it's the metal binding that's key. And I think excitingly, this is ready for application to other proteinopathies. For example, Parkinson's disease is an obvious one, or Alzheimer's disease where you get aggregation. So I think there's a lot of potential here for using this technology to reveal new biology about proteinopathies. So that leads me to acknowledge the Cooper Group, particularly Emma and James, who worked with me on this in the last couple of years. Very, very, very challenging work, very trying work. But when it comes together, it's fantastic. We've had really good collaboration with the University of Sheffield with mouse and human studies. Now, AstraZeneca have been fantastic, also providing tissue samples. And we work within the Advanced Mass Spectrometry facility at the University of Birmingham, but actually we're expanding to the Rosalind Franklin Institute near Oxford, near Didcot in the UK. And that's actually where I am right now. I'm in the booth there, and we're setting up a second lab there. And there's a postdoc opportunity if you're interested in applying for this. We've got a new mass spec, brilliant lab. So if you're interested, please apply. And yeah, good luck. Thank you.
Awesome. Thank you so much, Oliver. That was really a beautiful talk. And we've already got a lot of excellent questions in the chat. So participants, if you have questions for Ollie, you can either raise your hand or enter the question in the chat. I'm going to start with reading some of these questions. And of course, the people who wrote them can chime in to help clarify if I don't get the gist of it. And so Jeffrey Silva had some really, really great questions. So I want to start there. I think the first one that I think is we've gotten someone else also is asking something similar is, so SOD1, it can bind to copper or zinc. And so, you know, similar masses, do you have sufficient resolution to tell whether your metal bound forms are binding copper, zinc, both?
It's the $1 million question with this. And it's still not possible. There are some other experiments which we have planned which aim to solve this. My hunch with the 3 metal bound form is that it's going to be a mixture, because we see some of it forming, and it might just be some of that will be naturally mature SOD1. But so far we don't have a way of distinguishing between the copper and the zinc, and it really is the holy grail of this piece of work, I think.
Yes, certainly. And Jeffrey, yeah, if you want to sort of fill in that discussion a little bit, yeah.
Follow up on that. I'm curious to know if the formation of these deficient binders, do you think it's the result of oxidation or reduction of the metals?
I think it's probably something to do with the cellular environment where maybe there's a deficiency in the delivery of copper or zinc to the protein. So my understanding is with SOD1, it isn't known how the zinc is delivered. Whereas the copper, there's a known chaperone. And I think probably we're looking at some issue where the copper is delivered before the zinc and then the protein can't mature. That's my hunch. But there's a lot of contentious claims in the ALS literature regarding this. So it's extremely complicated.
So if I could, connect one more thing that you talked about was the glutathionylation of subtle. So, is that indicating a change in the, oxidative state of the cell? I mean, we're getting more reduction in formation of disulfides. Is that?
Yeah, I think
pointing
it's
towards
a
something?
really good question. So I think it's not outside the realms of possibility at all. We know at least some proportion of the metal deficient complexes do have an intact disulfide bond, which we can't say definitely one way or another that they all do, but we do know that there is a proportion of them with an intact disulfide bond as well. Yeah, the glutathione is a real big question as well, because it does indicate that there is something up with the cellular environment. If we look at the livers of these same mice, we don't see the glutathione in them. So it's specific to the brain that the glutathione is happening. So there's something up with the cellular environment.
The other thing I find,
And so, go ahead.
Sorry. The other thing I find really interesting is the localization in the spinal cord, how it nucleates at that in that area. Is there something unique to that area from a protein composition point of view?
So the ventral horn as an area is the disease pathology. It's where the motor neurons are. But at 120 days, there actually aren't that many motor neurons left in these models. And so I think probably that the spike in intensity we see, it's probably a motor neuron soma. And that's probably why we have that spike there. Yeah.
Awesome. I'd like to get to some of these other questions from other people. So Andrew Swanseger, you had your hand up. What question do you have for us?
Yeah, thanks. And thanks, Oliver, for the great talk. It's always wonderful hearing about your work.
Thanks,
I
Andrew.
noticed in one of your early slides, you mentioned that the tissue samples are unwashed, fresh, frozen. Is that correct? I'm wondering if you do anything to account for the, when you're doing protein imaging specifically, if you do anything to account for the glut of lipids that are going to come along for the ride, or if
yeah, the lipids are not something we solve on the sample. So what we do take them along for the ride, as you say, in the sampling and during ionization. But what we do is we use the ion optics on the front of the mass spec to eject them from the ion beam, essentially. So we are only transmitting ions with mass to charge above, say, 2000.
Okay,
Yeah.
so you're doing M over Z filtering with a quad or something.
Yeah, there's a little quad in the front of these Orbitraps, which you can use as a well, it's designed as a low pass filter, but it also kind of functions as a high pass filter. So so you can we use that initially, and then later stages of isolation as well. Yeah, to get rid of those.
Yeah, no, that's excellent. We've done that fairly frequently with a lot of heavier stuff with our work.
Yeah.
We've just noticed that when working with tissue samples, that the lipids seem to dominate the spray even prior to being able to do any of that filtration. So that's really interesting to hear that you can do that as well. Thank you.
Yeah, they're a nightmare. And if you don't have any of this filtration by mass to charge, you won't see any proteins. You will just see lipids. They are thousands of times more intense than the protein signals.
Yeah, precisely. All right, thank you.
Thank you, Andrew. We had a question from Paul Dennis, asking if you could comment on the time scale that it takes to acquire some of these data sets. Imaging
yes
can vary a lot. So tell us a little bit about that.
Yeah, so I'll start with, so if you're familiar with MALDI imaging, you'll know they do stuff like 50 pixels a second. We are more on the order of 1 second to 5 seconds per pixel. And this is because we need to do averaging of the spectra to build up the signal to noise ratio. And that's just part of the challenge at the moment with protein imaging, is you need to take a long time over it. Particularly, say, if you're on an Orbitrap system, you can average transients to really boost signal to noise. Spinal cord being small, it's fast. If you want to do the whole thing, it's only 2 hours. You can do multiple images in a day. That's quite good. The brains typically are 12 to 16 hours, depending on resolution and speed. Of course, the higher resolution you make it, the harder this is to run, and the longer you need to run it for, because you're requiring more pixels. And so that's why we make this compromise. You'll notice my high resolution images are all kind of targeted to a region. And that's because we've kind of done this scoping with lower resolution imaging to then go back to a serial section and target an image at higher resolution, just because of the time it takes to acquire them. Yeah, that's essentially the biggest problem. Yeah, the larger organs, it's one of the advantages of working with mouse as well. Mouse organs are smaller. So if you work with rat, your images take about 4 times longer.
Yeah. And so do you see any prospects for that and you know, improving? What's the path forward to making that more accessible?
Yeah, I mean, it'd be great to have better signal to noise ratio per scan. That if we could get per scan signal to noise ratio being boosted, and we're talking maybe an order of magnitude still, but sure. That would transform things because we could scan so much faster. And I mean, Julia Laskin's group for metabolites with nanodesi have done 500 micron per second imaging. That's fantastic. They've also got some amazing sparse imaging type workflows where where you don't sample the whole tissue and then you interpret it. I think that's fantastic as well. So there's all these extra tricks which can be used to speed things up, I think, too.
Sure, absolutely, Oliver. Eric, you had a question.
If I might chime in very quickly. Oliver mentioned the Laskin group. I'm one of the collaborators working on deconvolving this kind of higher or lower signal to noise level data, working with a Fourier transform deconvolution technique developed by the ProLab at the University of Oregon.
Oh, yeah, yeah.
So that's another way that you can sort of compensate for that lower signal averaging and higher noise baseline.
Are you doing that on the transients or on profile data?
On profile data.
Okay, yeah, that's
most of the time we don't have access to the transients. That is vendor classified information. So we're working directly from the mass spectra.
Okay, awesome.
We'll look forward to that work. Awesome. Eric, you had a question.
Yes, I mean, excellent thought. I really enjoyed. Thanks for your. I noticed you use C8E4, which is a non-ionic detergent for your controls with aquaporin.
Yeah.
But I also noticed, well, to my understanding, this is the first time at least that I see that 231 kilodalton proteins using MS imaging. Generally, relatively small molecules are identified. I'm wondering if you use non-ionic detergent to run all your experiments, no matter if you were interested or not in finding membrane proteins.
Yeah, generally now we include it always. It's very, very useful for setting up the experiments because it's a really good marker just for spray stability and stuff when we're setting up. But also it means we've always got the opportunity to look at lower solubility proteins. So we want to get as much as we can in there. And it just really helps particularly when you've got proteins with lipidations. They're soluble, but they're not soluble. You know, they're membrane interactors, and it helps solubilize them in particular. Yeah, that's the main reason.
Yeah, no, that's great. That's great. Thank you.
Sure. Well, I guess I had a question about whether you're also monitoring other metalloproteins while you're imaging the SOD1. I think we saw on one of your slides, I think we saw carbonic anhydrase. So, yeah. I mean, do you see changing in the extent of metallation of other metalloproteins?
No, not that we've noticed. And that's why we've kind of got this feeling that it's about the delivery of the metal ions to SOD1 specifically. And there's such extensive literature on this that I'm not so familiar with, but it's just a bit of a minefield. And that question about how the zinc is delivered, I think, is still a big one as well. But no, so far we don't see any metal binding that's weird. In the human cases, for example, we have S100B, and that combines zinc, and we see it with zinc and we see it without zinc, but we don't see any change across the tissue. It's just kind of consistent. But again, they're not SOD cases, so maybe those are not driven by a metal deficiency. Maybe they have a different mechanism. It's one of those things where I'm not convinced that all ALS is the same, and that could be because you get SOD1 proteinopathies with things like Parkinson's disease. There is a SOD1 proteopathy for it, and that just seems strange to me. So yeah, I'm not convinced that they're all going to be the same.
Interesting. Jeff, you had a question that you just put in chat, but maybe you can describe it a little bit more
Sure.
succinctly. Yeah.
Can you hear me?
We sure can.
I'm curious, as you're sampling the surface of the tissue slice, does the complexity of the sample at that position in terms of the number of constituent proteins affect what you're able to monitor?
In terms of things like overlap?
Yeah, so like if you're at a local position in the protein, the number of proteins is very high, as opposed to shift over and there's fewer proteins, and maybe the distribution of those proteins vary as well. How does that affect, what you're able to monitor? And yeah.
So one of the things we do is TIC normalization. So we should correct for any huge increases or decreases in total ion count across the tissue. The main culprit for kind of saturating the detector, so we're using an Orbitrap, so it's got a dynamic range of about 5,000. The main culprit is hemoglobin. So as soon as we hit a blood vessel, that's pretty much all you see. Luckily, the brain doesn't have very many big blood vessels, and so it's much less of an issue in the brain than any other organs. The reality of it is, though, that we don't actually hit our ion targets for almost any scan. So we're always sub-threshold of what we should be injecting into the analyzer anyway. So I don't think, especially with TIC normalization, I don't think we're affected by that. That may become a problem as the source becomes more sensitive.
Sure. Eric, did you have another question?
Well, I don't know. It might be a very naive question, but I don't have the structure of the. Can you hear me?
Yep.
Yes, we can.
Yes. Okay. I don't have the structure of CE4 in my mind, but I'm wondering, because you formed micelles, I'm wondering, once you dissociate the micelles from the protein, if you are taking metal ions with the non-ionic detergent as well. Something that might happen when you want to dissociate micelles forms with lipids, because the carboxylic acid group, right?
Yeah, it's not something we've seen, and it's something we were cautious of. So we don't see that. So when we're looking at metalloproteins, say for example carbonic anhydrase, we never see it without the zinc. It's always got the zinc. And the SOD1, we never ever see it with fewer than 2 metal ions in the dimer. If we were losing those metal ions, we'd expect to see them in a 0 and 1 metal binding form, and we've never seen that. We also looked for this because of concerns about collisional dissociation taking those metal ions, and it doesn't seem to matter until you dissociate the complex completely, and then you can actually rearrange the metal ions. But it's a very obvious pattern, and when we don't see that in the imaging at all.
Well, thanks.
No worries.
Very cool. Do we have any other questions from the audience? I had a question about how destructive the process is.
Yeah.
And so after you've rastered across a tissue sample, can you come back again and go across that same line? How much, relative signal are you going to see in that second time? Is it all gone? Are all the proteins gone?
So it's
or
somewhat
are you done?
dependent on the probe. It's one of the problems with nanodesi right now. Everybody who uses it has this issue. Is each probe is different. Each probe has its own character, and some of them are fantastic and will last you months, and they don't destroy the tissue, and they're just absolutely amazing. And then you put the next one on, and it will vacuum your tissue into the mass spec, and you can't do anything afterwards, and the signal's horrible. So there's that variability with probe is a real pain. So if you have a good probe, yeah, you can go back over the sample. The signal intensity of everything is reduced, but you can still analyze. At lower signal intensity, what was there. Yeah, it's it's one of those things that would be really nice to be able to do an image and then go back and do all the MSMS in the same place. But I know, for example, it was Pei Su, wasn't it, who developed the the MSMS. So you do the line scan and then you do the MSMS along the subsequent line scan. I think that's a really neat solution, and I think that's probably the way to go for an automated type top down.
Well, sure. And I was, we've also seen examples of trying to study multiple analytes
Yeah.
on the same tissue. Yeah.
Yeah.
I don't know if you guys have thought about trying to combine to beyond just native proteins.
Yeah, I mean, that would be fantastic. I think one of the concerns is the disruption of the localization of any, say, lipids, for example, as we go over. One of the things I like the idea of is having, so if we had 2 high resolution mass analyzers in the same system, we could inject the proteins and the lipids by optimizing the ion optics at the front end, and we could image both at the same time. We can do that on the ion trap of the Orbitrap, but of the Orbitrap Eclipse platform, say. But lipids really do require high resolution for imaging, and so proof of concept works, but it's it leaves a lot to be desired. I think as well, we mustn't forget about other techniques, like other microscopy techniques, fluorescence imaging and that kind of thing, because these are all additional information which, if they can be got either before or after imaging, that would be fantastic. I think practically before is the way to go, though.
Sure. All right. Well, thank you so much. It was a beautiful talk, and I think we all learned a lot. So let's thank Ollie one more time, and I'll wish you all to have a great rest of your day.